next energy system, or the next ATP producing system we're going to talk about, is the electron transport chain. But before we can go into that, we need to go into a little bit of background. So remember when we talked about glycolysis, I said there was some NADH plus H plus produced that went to the electron transport chain. When we talked about the intermediate step, there's NADH plus H plus that I said goes to electron transport chain. We've also got some NADH plus H plus in the Krebs cycle, which goes to the electron transport chain, and some FADH2, which goes to the electron transport chain. But we haven't talked yet about what these things are. What are these NADH plus H? What is this FADH2? So before we can talk about the electron transport chain, let's talk about that. So FAD and NAD are coenzymes. Okay, so we have two different types of coenzymes, and they're responsible for picking up hydrogen ions that are released. Okay, and hydrogen ions are released in chemical reactions, and as you can see, or as you have seen, we've got some hydrogen ions in glycolysis, in the intermediate step, in Krebs, and as you will see, we also have some substrates releasing hydrogen ions in beta oxidation. Whenever that happens, hydrogen ions are picked up by a coenzyme, either by NAD or FAD. And the role of NAD and FAD is to pick up those hydrogens, carry them to the electron transport chain. Once we get there, we can talk about what goes on in the electron transport chain. Before that, I want you to also know that wherever there are electrons, there are also hydrogens. So, or I should say, wherever there's hydrogens, there's also electrons. So I keep talking about hydrogens being released, hydrogens being picked up. But keep in mind that simultaneously with the hydrogens come electrons. So if hydrogens are released, electrons are also released. If hydrogens are picked up, electrons are also picked up. So NAD and FAD are coenzymes. You may know them by their more common name. Nicotinamide adenine dinucleotide is also called niacin, which is vitamin B3. And flavin adenine dinucleotide is also known as riboflavin, which is vitamin B2. So those are carrier molecules. They participate in the process of making ATP because they have the important role of carrying hydrogens and electrons to the electron transport chain. And as you will see, that's where we get most of our ATP. So let's talk a little bit more about NAD and FAD and how they work. So we have a process called reduction and oxidation. So reduction means that some carrier, okay, and our carrier is either going to be NAD or FAD, accepts hydrogens and electrons, okay, and becomes reduced. So when any substrate takes electrons from another substrate, we say that that substrate becomes reduced. Okay? So at some point, NAD picks up hydrogens and electrons, becomes NADH plus H plus, and remember there's also two electrons there. And therefore, at this point, we will say that NAD is reduced, and its reduced form is NADH plus H plus. Okay. FAD does the same thing. It accepts electrons and hydrogens. You can see it also picks up two hydrogens, which means it picks up two electrons. The difference is that FAD binds both hydrogens, So we write it as FADH2 instead of NADH plus H plus, just a slight difference, and again, we're not going to go into the chemistry of that. So when FAD is the acceptor, FAD becomes reduced, and its reduced form is FADH2. Now, in the reduced form, okay, these reduced molecules are very energy rich because the electrons that they carry potential energy. So basically what we're doing is we're picking up hydrogens and electrons, carrying it to the electron transport chain. In the process of the electron transport chain, we're going to take this potential energy out of electrons and use it to make ATP. So we call this reduction. So to summarize, when something is reduced, it gains or accepts electrons, okay? And remember that the hydrogens also come along. And so we will say that the electron acceptor accepts or gains electrons and becomes reduced, okay? So when NAD picks up 2 hydrogens, 2 electrons, it becomes NADH plus H plus. We don't write the electrons, but know that they're there. Okay, so NAD accepted electrons and became reduced. Okay, and this is half of a redox reaction. So redox is reduction, oxidation. Okay, so reduction is always accompanied by oxidation. Okay, so we've got reduction going on, and therefore they always are accompanying each other, so oxidation must be also going on. Let's talk about what oxidation is. Okay, so oxidation is the act of losing electrons. Okay, so on the last slide I said that NAD picked up some hydrogens. Okay, well, those hydrogens and electrons had to come from somewhere. Okay, and it came from this other substrate. Okay, so in this example, X lost electrons. X was the electron donor. Okay, X gave its electrons and hydrogens to NAD. Okay, NAD was reduced. X was oxidized. Okay, so the substrate that donates its electrons becomes oxidized. So it's the supplier of electrons. It loses electrons. And in the process, it becomes oxidized. So we have a redox reaction. And you might have heard oil rig. This is a way to help you remember what is being oxidized, what is being reduced. So oil rig, this is oxidation, is losing. Okay, so oxidation is losing. So X lost. Reduction is gaining. The NAD gained the hydrogens and electrons. Okay, so in a redox reaction, you've got one substrate being oxidized, another substrate being reduced. And together we have a redox reaction because the hydrogens and electrons had to originate somewhere, originates with the donor, and it had to go somewhere, goes to the acceptor. The donor is oxidized, the acceptor is reduced. Okay, so whenever we have these going on, let's look a little more carefully at this. I told you don't need to know the names of the substrates, and you don't. But in this example, let's just say this is substrate A, 3-phosphoglyceraldehyde, and this is substrate B, 1,3-diphosphoglycerate. Okay, so what happened here was substrate A gave its hydrogens and electrons to NADH plus H. Okay? So substrate A donated its electrons. Substrate A lost its electrons. So substrate A was oxidized. Those hydrogens and electrons were picked up by NAD. So NAD becomes reduced. And what we end up with is substrate B. Okay. So substrate A was oxidized, NAD was reduced. The result is substrate B. And now that NAD can carry those hydrogens and electrons off to the electron transport chain. Okay. And remember that this happens twice. We've got a 2 here, because this glucose 6-phosphate divides into 2 3-phosphoglyceraldehydes. So this happens twice. We get 2 NADs that go off to electron transport chain. Okay, we also see some redox occurring in the intermediate step. So in the intermediate step, pyruvate gets oxidized, gives its hydrogens and electrons to NAD. NAD gets reduced, carries them off to ETC. If you look at the Krebs cycle, isocitrate gets oxidized, alpha-ketoglutarate gets oxidized, malate gets oxidized. Okay, they all go off to ETC. And then succinate also gets oxidized. The only difference here is that the carrier of the hydrogens and electrons is FAD, not NAD. Okay, so succinate is oxidized, but instead of those hydrogens and electrons being picked up by NAD, they're picked up by FAD. So in this case, FAD is reduced. Okay, and again, carries them off to the electron transport chain. And that's what we're going to talk about next. What happens when FAD and NAD arrive at the electron transport chain with those electron and hydrogen pairs? Okay, so electron transport chain, sometimes referred to as oxidative phosphorylation. Okay, so we've got some oxidation occurring. Remember that oxidation is losing. So something is losing electrons and hydrogens. Okay? And we've got phosphorylation going on, which means something is gaining a phosphate. And since we're talking about energy metabolism, it's probably ADP that's being phosphorylated. Okay, if we bind a phosphate to an ADP, we end up with ATP, and remember that's our ultimate goal. So where's this energy coming from? I told you that these electrons carry potential energy. So what happens in the electron transport chain is that the potential energy is released from the electrons and used to phosphorylate, to add a phosphate to an ADP, and what we end up doing is making ATP, which is our ultimate goal. It's an aerobic system, so it occurs in the mitochondria, and the function is to make ATP using energy from electrons. Okay, and those electrons originated in glycolysis, Krebs, intermediate step, and as you'll see later, beta oxidation. So we've got hydrogens and electrons being released all over the place, carried to the electron transport chain. Within the electron transport chain, we get some oxidation, some phosphorylation, and therefore we produce ATP. All right, so electron transport chain begins with either NAD, H plus H plus, or FADH2. So we're going to go through it twice. We're going to go through it with starting with NAD as the carrier, and we're going to go through it starting with FAD as the carrier. It's a very slight difference, but you need to know the difference. The first thing that happens in the electron transport chain is that NAD and FAD are oxidized. Okay, remember oil rig. Oxidation is losing. So NAD and FAD lose their hydrogens and electrons. Okay? And remember, the hydrogens and electrons are always together. The next thing that happens is that these hydrogens and electrons split. You'll see it on the next slide. The electrons get passed down a chain of cytochromes, and as they're passed down the chain, the cytochromes are reduced, okay? Because the cytochromes are accepting and gaining the electrons, okay? Gaining is reduction. So as the cytochromes pick up the electrons, they become reduced, and as they pass them on to the next cytochrome, they become oxidized. Oxidation is losing, so as they lose their electrons, they become oxidized, and this happens one by one. And in the process, as these electrons are carried along, transported and passed along the cytochromes, they liberate energy. Okay? And that energy is used to bind a phosphate to an ADP, and we end up producing ATP. Okay, so let's see what this looks like. So first time through, we're going to talk about NAD, and what happens when NAD is the carrier. So cross this out. Okay. So what we have here is we have NAD arriving, carrying 2 hydrogens, 2 electrons. Could have come from glycolysis, intermediate step, Krebs cycle, or beta oxidation. And it drops off its electrons and hydrogens. It loses them. Okay, so it gets oxidized. And what you're left with is NAD. That NAD can go back to glycolysis, intermediate step, Krebs cycle, or beta oxidation and pick up more. So it's just a transporter. Carries them over, drops them off, goes back to get more. Okay? So these hydrogens and electrons are split, and now the electrons get passed down a chain of cytochromes. So we have several cytochromes. Okay. And so the first cytochrome accepts the electrons. Okay. So gains the electrons. So cytochrome A gets reduced and then passes the electrons on to cytochrome B. Cytochrome B gets reduced, and then A gets oxidized. B sends it off to C, and now B is oxidized, C is reduced, etc. Okay, so you're going to go on down the line until the electrons end up at cytochrome E. And in the very last step of electron transport chain, we have a half of an oxygen molecule. So remember, oxygen is O. So half is O2. So half of an O2 is an O. So we have half of an oxygen that comes in. It accepts the electrons. The hydrogens come back. So we have an O, we have 2 hydrogens, we have 2 electrons, and we end up with H2O, water. Okay, so the role of oxygen here is that it was the final electron acceptor. Okay. So cytochrome, these cytochromes accepted the electrons, passed them on and on and on and on, and at the very end of the line, the last thing to accept the electrons is oxygen, and that's where the electrons stay. So the half of the oxygen, the 2 electrons, the 2 hydrogens all come together, and we end up creating water. Okay? So we call oxygen the final electron acceptor. Also note that as we went down the line, we produced some ATP. Okay? So because these electrons had carried energy, and so the energy from the electrons were used to phosphorylate ADP, and therefore at the end we end up with 1 water, 3 ATP. Okay? And an NAD, which goes off and gets more. Okay, so we start with an NADH plus H plus. We end up with 3 ATPs, 1 water, and an NAD that goes back and gets recycled and picks up more electrons. Okay, so that is the process of electron transport chain when the carrier is NAD. When the carrier is FAD, cross this out. Okay, the process is identical. Oxygen is the final electron acceptor. We end up with a water. The electrons and the hydrogens come together. The electrons were passed on down the line. The only difference is that FAD enters the electron chain in a different location, and therefore we only get 2 ATP when we start with FAD. Okay? So everything else is identical. FAD goes back, picks up more hydrogens and electrons. Okay, so we started with FADH2. We end up with 1 water, 2 ATP, and that FAD, which goes on to get more hydrogens and electrons and bring them back. All right, so at the end of the electron transport chain, oxygen is the final electron acceptor. Oxygen is the very last thing that gets reduced. Okay. Reduction is gaining. So oxygen accepts the electrons, gains the electrons, becomes reduced. And then the hydrogens and electrons combine with the half of oxygen forms water. Okay? In the process with NADH plus H plus, we get 3 ATPs from FADH2, comes in at a later step, so we get 2 ATPs. But the actual net gain, and these are the numbers you need to remember, the actual net gain is actually 2.5 and 1.5, because in the process we use a half of an ATP. Okay, so our actual net gain is 2.5 and 1.5, depending on where what carried those hydrogen and electron ions in. Okay. And that's the electron transport chain.